So these specimens are the basis for herpetological research. The research itself is important because anything, whether it's ecology, phylogeny, morphology, all that stuff goes into informing conservation decisions on these species. If you don't know anything about the animal, you can't really figure out how to protect it. There's two different initiation points. One is we have a dead frozen specimen, one is we have a live specimen. With a live specimen, we'll euthanize it in the prep lab there, take our measurements, so body length, tail length, weight, anything else of note on the particular species. First thing to do is assign it a specimen number. So we have a list of tags, cut off a tag from that, place it with the specimen, find the corresponding tissue vial with the same number as the tag, and after that we can begin taking the tissue sample. So to do that, we make an incision between the ventral scales for a snake. For frogs and lizards, it'll be kind of on the chest, just below the rib cage. Take a small chunk of liver, no bigger than like pencil eraser size, place that in the tissue sample vial with a preservative called RNA later, and it's essentially salt water that'll go into cold storage and prevent degradation of the nucleic acid, so that sample can be used for genetic analysis far in the future. Tie the tag onto the specimen, so for a lizard or a frog or a turtle, that'll get tied onto the hind leg. For exceptionally tiny specimens, it'll go around the waist, and for a snake, we use a needle to put the tag through the specimen. Then we'll take it back to the fume hood to fix it. For fixing, we use a compound called formalin. It's dilute formaldehyde, and that both prevents decay. No bacteria can grow in formalin. It's extremely toxic, so nothing's able to decay the snake, and it also helps the specimen keep its shape. So if you look at the jars around here, all the snakes are coiled up and they'll stay in this position. They can move around a bit to be pulled out of the jar and to be measured and whatnot. The process there is just to inject down the body with formalin. Don't gotta go crazy with it because formalin will penetrate the tissues on its own. So that's why we use formalin instead of ethanol, which is the historic way of doing it. Then we leave the specimen in a covered container with paper towels soaked in formalin for 24 hours. That'll give it time to harden. After that, it's moved to a jar of formalin, and over a period of time, we'll progressively dilute the formalin and eventually replace it with ethanol, which is what is used for permanent storage here.